Hi everyone and welcome. It's a pleasure to be back after my short sabbatical, and it's a pleasure to introduce the lecture. And I would like to thank Marcus for providing us the opportunity to have Florian with us. Dr. Florian Heidel, he had academic formation in Germany, in different places in Germany, in the medicine area, in Jena, and other places there, where he did his medical training. He did later a postdoctoral stay with Scott Armstrong. Scott Armstrong is one of the, in my opinion, one of the fathers of epigenetic translation treatments in leukemia. He's a great scientist. He stayed there at least 5 years or so, more or less. And he went back then to Germany, first as junior research group leader, and later now became a deputy director in Jena, and now he's the director and chair of the internal medicine, hematology, oncology, stem cell transplantation in the Strayed in Greifswald. Okay, Strayed. And he's a world expert in hematological malignancies, mainly in the area of downstream effects of JAK really in the myeloid lineage. He has made very important contributions, and I think that, just talking before, he's going to make even more contributions in the coming years. So it's really a pleasure to have with us. He's a partner of Carreras Foundation, being granted by the German Foundation. So really he's a friend and collaborator of the Institute. So Florian, great having you here.
Thanks so much, Manuel. Thanks, Marcus, for the kind invitation. Happy to be here and being able to speak. Topic of my talk today will be cell signaling. And JAK-dependent cell signaling is something that is in our focus of interest. But let me start a bit. As you can see, I'm still affiliated with Leibniz Institute on Aging. So there is an aging focus on that, and why is aging connected to JAK mutations? I would start with the bigger challenge that all of us in the Western world at least face, and that is demographic change. And of course I start with German data, but I think in every Western country it's more or less the same. If you look at the distribution of the population in 1990, you can see the majority of people are in the third or fourth decade, and that massively changes. If you look what we can expect in 2030, the majority of the population will be in the 60s or 70s. And that aging is region dependent, at least in Germany it's region dependent. You can see there are some spaces, some areas where the aging is more prevalent than in others. But overall, you can see 1/5 to 1/3 of the population is 60 or beyond. So what comes with aging is aging of the hematopoietic system, and this is my primary interest as a hematologist, also clinical hematologist. And stem cell aging comes with different properties, as most of you know. This is decreased regenerative potential. Hematopoiesis can still react to extrinsic stimuli, irradiation, chemotherapy, etc., but the regenerative capacity is usually lower. There's myeloid skewing, which means that has been shown in mice, but it's also present in men. With age comes a predominance of the myeloid lineage. Then increase, and this is, I want to say, clearly immunophenotypic increase of the HSC pool. If you have an old mouse compared to younger mouse, the old mouse has more LSK cells, more stem and progenitor cells immunophenotypically. However, if you do functional assays with these cells, you will notice that function declines, although they are more. And finally, and this relates mainly to the human system, what we know is that age-associated mutations appear in the peripheral blood. They are detectable, and this had great impact in the last 7 years or so, starting with the observations of Ben Ebert and others. who discovered that even in the healthy population, so healthy individuals, not patients, harbor mutations that we normally know or that we initially knew from hematopoietic cancers. And among them is DNMT3A, TAT2, ASXL1, P53, JAK2 as a kinase mutation, activating SF3B1 and other signaling proteins. And this is frequent, you can see, and the frequency increases. And this is the numbers that Benibet put up at the initial publication. If you now look in more depth and you look in higher numbers, that may even be detected at a higher number earlier. Even in the 40s or 50s, you can detect these mutations at lower levels. So question arises, what are these mutations doing? And the good answer is, the good part of the answer is not all of these individuals will develop blood cancer, but the risk of blood cancer is increased if these mutations are detected. But what is, from my opinion, even the bigger threat is the effect that this clonal hematopoiesis, as it's called, has on other diseases, cardiovascular diseases, for example. That means ischemic heart disease, that means stroke, arteriosclerosis. And there's this interplay between clonal changes, myeloid malignancies, and cardiovascular diseases, but the threat may be even bigger on these very frequent diseases and the morbidity and mortality that comes with it. So speaking about that, we have a continuum that I wanted to highlight. The continuum is that you may have this clonal hematopoiesis of indeterminate potential. That means you have a mutation, but you don't know if that just stays stable over time, if it disappears, the clone, or if it expands and becomes something meaningful. Then you have something intermediate that is called CCAS mutation there, but you have changes in hematopoiesis, for example, not otherwise explained anemia. And then there is pre-leukemic chronic neoplasms. This is what we especially, it's a shared interest with Manel, and we have this MDS and MPN phenotype, that means either dysplastic and loss of function or hyperproliferation, causing problems also because of hypercellularity. And JAK mutation, and that may even progress to overt acute leukemia. So JAK mutations, and I started with JAK, they are detectable in all of these stages, in CHIP and CCAS, but they are very frequent in the proliferative part, especially in MPN, where they are even called driver mutations. Now, I would like to spend some time to talk about this aspect of driver mutations, because one of the reasons why I am so interested about JAK signaling is they're a bit different than others. So what you know, what is textbook knowledge is that there is the wild type JAK receptor, and JAK is responsible for mediating cell signaling at receptors where the receptor itself doesn't have kinase activity. Usually ligand spined, and these ligands are highly variable. It can be interleukin receptors, it can be hormone receptors like TPO or EPO receptor that are responsible for hematopoiesis, and then it's activating of mainly STAT-dependent signaling. And textbook, if you read textbooks, it's mainly JAK-STAT signaling that is pronounced in this way. But it's more than that, because there is interaction with other pathways. There's MAG-ERG signaling, PI3 kinase AKT, still very limited. What is not known so far is what is the network of signaling below activated JAK kinase, and this is what we became interested in. And we became interested because there is a side of activating. There are also inactivating JAK mutations that may cause resistance in other types of cancer, but I will stay on the myeloid neoplasm or clonal hematopoiesis side, where activating JAK mutations lead to activation of all of these pathways. And And what we are interested in is this purplish thing here. What are unknown effectors and what are unknown interactors of these signaling pathways? How are they maintained? And one last thing before I start with the data. The reason why this is so important is that JAK kinase has two things that are different from other kinase mutations. First, left side, this is a waterfall plot, data from Michael Deininger in Blood, 2015. And what he shows is responses, molecular responses. So this is JAK2 allelic burden in the peripheral blood of patients treated with a JAK inhibitor, ruxolitinib, for a long time, over many years. And what you can see is, so first of all, this is a linear scale here. You can see that 80% of the patients have minor or no response, not even 50% reduction of the clone. Only 20% do. And this is a clear minority. Let's take CML and BCR-ABL. Then this axis would be logarithmic, and 90% of the patients would reach a log-fold reduction. Same thing for other kinase mutations, but here you see, although you inhibit the kinase, the clone persists. Second thing, and this is a very recent Nature paper from George Vassiliou's group, where they traced clonal aberrations over time and described that depending on age, these mutations have mostly linear kinetics. Some are influenced by age, some are not. But JAK mutations have been highlighted to be different because they do not follow a linear expansion that is either age dependent or not. You can say, see, this is completely erratic. Some of them expand and make disease, some are stable over years and decades, and others even disappear. So it's completely unclear what influences this, and also it seems to be not related to the activity of the kinase. So how can one approach this field? And we sought to team up with the group of Matthias Mann at the Max Planck in Munich to have a broader look, a global view, as we do with transcriptomics for many years, a phosphoproteomics approach to understand what the signaling network is downstream of JAK2. And in very brief, we used a very standardized murine model where you have a defined receptor that is stimulated, and you have a defined introduction of JAK kinase, either wild type or active mutant, and you can, by this way, stimulate the receptor in the wild type and mutated stimulation, and you can also inhibit it like drug perturbation by a JAK inhibitor. And this was followed then by a phosphoproteomic analysis. Now, one more word before we go to the results. What you get out is you will see a high number of proteins that show changes in phosphorylation at different residues. Just to be clear from the very beginning, we are not talking about one phosphosite, like in Western blotting. It's rather that one protein, here one example of the protein that we studied in this study, has dozens of phosphorylation sites, and they may be differentially regulated in the wild type situation versus the mutant situation. So this is that, and a change in one specific phosphorylation site does not mean necessarily it has a functional impact. So to the data. What we found in the first place when investigating this model system of activated JAK2 kinase was a massive amount of phosphorylation changes. We're talking about more than 6,000 phosphosites correlating to over 1,500 proteins. That shows some degree of differential phosphorylation or significantly dephosphorylated or phosphorylated. The first important thing was, do we find targets that we know about? And the question is yes. So you find the stats, you find PIM kinase, here EPO receptor, STAT5. So the bona fide targets are there, which is an important validation step. But beyond that, we found proteins that we didn't know about. They are regulated by JAK, and second, also cellular functions that we did not expect. And one cellular function that was at that point really unexpected was RNA splicing and processing to be regulated by JAK-dependent signaling. So this was one aspect that we really gained interest in, and you can see those are the top 15 proteins that have been differentially phosphorylated downstream of JAK2. You find, first of all, splicing factors, SRSF2, for example, that are, or SF3B1, that are not only differentially phosphorylated, as we have shown here, but are mutated in the same type of hematopoietic cancers. So they seem to be regulated by, or there seems to be a pressure in the direction of acquiring these mutations, but obviously they are also target of the kinase itself. So what we sought to do is a functional characterization, first in vitro, by inactivating these factors and see what they do to these cells in a dish. Do they inhibit proliferation and viability? And this is the result in very brief of this screen. It's hRNA-based screen at that time. And you can see here growth of the cells. And on the left side, this is inactivating these 15 genes with at least 4 shRNAs per target. And on the right side, it's the same infection with the same pool of guides, but co-treatment with a JAK inhibitor. So what happens here on the left side, if a cell drops out, or if a clone drops out that is inactivated, it's clearly dependent on the protein or gene. On the right side, it's basically the synergy with the co-treatment of JAK inhibitor. So this is pure dependence, and this is synergy, if you will. And one of these targets that gained our interest was YBX1. As you can see, 4 out of 4 dispensable for the cells in the situation where you have no treatment, where the kinase is active, but it gains obviously massively importance when you inhibit JAK kinase. And this is the setting that we want to understand, right? The inhibitor doesn't eliminate the clone, but obviously if you find another regulator that is important, you may be able to reduce or even eliminate the clone. So what is YBX1 doing? It's a pleiotropic protein, first of all, which doesn't make it easier to study. It has been described as a transcription factor to be involved in mRNA stability, to be a signaling effector being phosphorylated at different residues, has been described to interact with epigenetic modifiers, and that has been also investigated, all of these effects in different tumor models. So it seems to have context-dependent roles, and this is the important thing. And in our model system, in diseases that carry JAK2 activating mutations, first of all, and this is what a clinician wants to look at also, is it there, is it expressed? And you can see if there is an activated JAK kinase, you have high expression of YBX1, predominantly in megakaryocytes, but also in immature erythropoiesis and granulopoiesis. So what does it do functionally? We went into cell line models and tried to use different cell line models that have all an active JAK2 mutation. So we used mouse cell lines, human cell lines with a JAK-VF mutation, and primary mouse cells from a JAK2-VF knock-in mouse that we extracted from the bone marrow and then treated in vitro. And what you can see here is similar to that screen. The inactivation of YBX1 alone has, if at all, minor effects on cell viability. However, when you combine it with JAK inhibition, you see a significant increase in apoptotic cells, and this is consistent with human cell lines, and also consistent in the case that it's not a cell line, but a primary cell out of a mouse. And that shifts, obviously, the sensitivity to to a degree from more than almost 2 micromolar than to a nanomolar range. Now this is all cell culture models, and what we know from the signaling field is if you keep investigating these cells in a dish, you may miss signals from the outside, from the environment that may heavily impact the phenotype. So we clearly wanted to investigate that in vivo, and we created here a mouse model in which exon 3 is targeted, and targeting this exon by active recombinase leads to complete loss of a functional protein. So YBX1 is completely deleted, and we used this model system to intercross it with a mouse model that has a conditional knock-in of this JAK2 mutation, activating JAK2 mutation. So what you could do here is, by activating recombinase at the same time, knock in the JAK2 mutation, at the same time inactivate YBX1 on both alleles. To see what happens with these cells, we used a competitive assay where you have 50% of competitor cells and 50% of the cell population you want to study. So what you look at in the end is a chimerism. That means proportion of test cells to competitor cells. You can really see how the competition level is over time in vivo. And the results are here. So let me guide you through that. On the left side, this is blood counts. In dark gray, you see what happens if you knock in a JAK mutation. It leads to an increase in white blood cell count here over time. It expands in the chimerism, and it does so even if you treat with a JAK inhibitor. And this is completely consistent with what we've seen with Michael Daninger's data in the patient. However, if you inactivate YBX1 via the recombinase, that does not happen anymore. You see the white blood counts stay stable, hemoglobin even decreases over time, and also platelet counts stay down. And most importantly, when you look at the chimerism, these cells get outcompeted. So this is a loss. This is a logarithmic scale. So if you assume a 1-to-1 ratio is 50%, then we are here in the range of 1%, and some even disappear. They are completely outcompeted, which is usually not the case. So you have a loss in more than 50% of mice. Now again, validation in a human system would be warranted, and one first approach, and I agree this is not an optimal setting of the experiment, was to inactivate YBX1 or non-targeting, or use non-targeting control in a mouse model where we inject human HALE cells. HALEs carry JAK2 mutations, but, and this is the limitation, it's a model system where this disease has already converted to leukemia. So it's a leukemic JAK2 mutated type. We injected them into mice and then treated either with saline or the JAK inhibitor, and the result is somehow similar. What do you see if you inactivate this YBX1 by a lentiviral transduction of the shRNA is a minor delay. However, if you co-treat only for a short period of time with a JAK inhibitor by gavaging the mice, you see a little increase in survival in the only JAK inhibitor treated group compared to this, but the difference of the YBX1 inactivated is much larger, and you see some even don't get sick for 100 days and more. Now we have identified a target. One thing that we would be really afraid of is, with every therapy, every approach to eliminate a clone, toxicity. And JAK2 signaling, I told you, EPO receptor, thrombopoietin receptor signaling, interleukin receptor signaling, all dependent on JAK. What happens to normal hematopoiesis if you inactivate such a gene in the mouse? So we did that whole experiment without the presence of the JAK mutation, just to see what does YBX1 deletion do on its own, and it's in a way not affecting massively normal hematopoiesis. There's no significant change. There is no significant change in immature populations of the bone marrow, neither progenitor cells nor hematopoietic stem cells. And you can see here the numbers. That is pretty much unchanged. And also, if you take these cells out of the knockout mouse and you transplant them into primary hosts, you let them expand for 6 months, you take them out again, you retransplant for a second time. So you stress them, you bring them in cycle again, and this is what I show you on the next slide. This is a 50% chimerism here, the dashed line, and you can see knockout in green versus wild type in black, and this is basically no difference over a year of retransplantation experiments. So let's go back to the mutated version. And of course, we asked the question, what is now the mechanism? If you inactivate this protein, YBX1, what happens? First of all, on the transcriptional level, and the first thing that we found made sense in a way, that the inflammatory response, the cytokine production, hallmarks of activated JAK2 inflammatory signaling is blunted, and also MAP kinase, interestingly, and ERK signaling was affected among the most significant functions that was deregulated or signaling pathways. And when we looked a bit in more depth, we found that there was differential splicing that was significantly altered. Actually, intron retention was one of the major effects that could be found. And if you look a bit more in that group of genes that shows intron retention, then there are indeed MEK-ERK, MAP kinase signaling related genes that show a significant degree of intron retention. And the question was now, out of these, and I show you 3 of them, which ones show also a real effect on protein abundance? And that was pretty clear. There is MKNK1, a stabilizer of MAP kinase signaling that showed in both proteomics analysis, and here is the control of Western blotting, again in mouse cells. In human cells, you see inactivation of YBX1 leads to a depletion and even abrogation of MKNK1 expression. So this may be related to the differential splicing event. If this is so, first question was, can we perturb the spliceosome machinery by pharmacologic perturbation? And do we see the same effect on the signaling protein? This is what we did with 2 different splicing inhibitors, inhibitors of the spliceosome machinery, platelinolid and herboxidine. And in both cases, you can see there is a reduction of MKNK1 abundance in JAK-mutated cells, as here as one example assessed by flow cytometry. Now, the next question was, if this is really splicing and this is a protein that is phosphorylated at different sites. First of all, which sites are responsible for this phenotype, and what are these phosphosites doing, if so? So one thing that I can't show you for time reasons is that we have done a pharmacologic perturbation screen and could identify residues that were different, first of all, differentially phosphorylated, and second, also conserved between species. And among them were the position 30, 34, 172, 174. And what we then did was to create mutants, phosphomutants, mutating either to A or to D, and expressing these mutants in cells with a JAK2 mutation. And what you can see is there is indeed, first of all, a difference in nuclear localization, which may indicate that these phosphorylation sites are relevant for nuclear transport to exert the function. And we could see most compromised were S30 and 34. And this was not only visible in microscopy, it had also a functional readout. Those perturbations or mutations of the phosphosites led to decreased proliferation, especially in the presence of JAK inhibitor treatment. So we did more experiments to consolidate that. I will just briefly tell you. So we also investigated transport proteins that transportin that is responsible for translocating YBX1 to the nucleus. We mutated the binding sites. Even with mutation of these binding sites, you see the same phenotype regarding nuclear translocation and also effects on cellular proliferation. So taken together, until now, YBX1 seems to be responsible for alternative splicing. It seems to affect genes that are necessary and responsible for maintaining MEK-ERK signaling in JAK-mutated cells. And responsible for this effect is obviously phosphorylation at conserved amino acid residues that are responsible for nuclear translocation and obviously also for interaction with the splicing machinery. So what we thought is going on is that there must be some stabilization of ERK signaling unless you inactivate YBX1. And that is one experiment that we did to do a time course and see, even if you use a JAK inhibitor on JAK-mutated cells, what happens to the ERK signal. And there's a weak ERK signal that is primarily on. You see it goes off when you use a JAK inhibitor, but it comes back on early. And at 96 hours, it's massively back on, even in the presence of JAK inhibitor treatment. So there's a rebound, and this rebound obviously involves ERK substrates, and MK1 was in proteome analysis after inactivation of YBX1 along with MCL1 among the mediators that we got into focus, and that confirms the results from RNA-seq analysis and intron retention. So to confirm this, and this is Western blotting, this is intracellular flow, you can see exactly that in the wild type setting, in the untreated setting, I'm sorry, this is all JAK-mutated cells. You see when you inactivate YBX1 by RNAi, there is a decrease of ERK signaling. Likewise, if you use a JAK inhibitor, you get also a reduction of the signal. This is what you see here. This is the same effect here. However, if you combine both, you abrogate the signal, and this may be enough to induce apoptosis and kill the cell. And this can be confirmed. This is mouse cells. This can be confirmed in human cell lines, but also we did that by flow in primary patient cells. In those selected with a high mutation load, 88% of JAK2-VF mutational burden, so the majority of cells investigated here. The knockdown leads to a reduction of the ERK signal. And interestingly, and this is now the translational angle, that is also achievable if you do not inactivate YBX1, which you can't do in a patient, but also inhibiting the downstream effectors. So using a MEK-ERK inhibitor that blunts the signal that comes back on. If you can provide a pharmacologic solution for this, it would be inhibiting MEK-ERK signaling at this point, so the rebound does not happen in the cell apoptosis. Does that work? It does. This is cell line data. Let me walk you through. We did that with inhibitors of the intermediate, so of MK and K1. You see induction of apoptosis. We did that with the JAK inhibitor ruxolitinib and a MEK-ERK inhibitor, trametinib. It's a drug that is approved for treatment of melanoma in the clinic, so it's available. Same thing, induction of apoptosis. Now you could say, well, you did all of that only with one JAK inhibitor. This may be just an effect of this very drug. It's not. It's a class effect. You can see even using other JAK inhibitors, pacritinib, same effect, induction of apoptosis. So it's an effect that can be seen in general. And now the criticism could be it's all cell lines. True. You can see the same thing in primary patient cells. So this is JAK mutated cells out of the patient. This is control cells mobilized 34 from healthy individuals. You can see neither treatment with JAK inhibitor nor the combination does anything to normal 34 cells in terms of viability. You can see that the combination compared to Rux alone, either inhibitor, JAK inhibitor Rux or pacritinib, increases massively the amount of apoptotic cells. Now, it's still at a point where we cannot do preclinical assessment. To bring it into a trial, what you would like to have is an in vivo system where you can try that out either with genetic or pharmacologic perturbation. Now, the difficulty is people in the field of you who work with xenograft models know how difficult it is to engraft leukemias sometimes. So it's even more difficult to engraft pre-leukemias, MDS, MPN, that's super difficult. Low engraftment rates. And if something engrafts, it's sometimes a progression of what you injected. So we teamed up at this time with Claudia Vasco, who's a long-term collaborator and of ours at the Leibniz Institute. And what she did, she generated for investigating the human immune system in mice. She generated a model of NSG mice with a mutation of KIT at the position W41. So I refer to these mice as W41 mice. And you can engraft primary human cells without any mutation at high levels in this mouse model. It's very permissive. So we thought if she can engraft normal cells at high levels, we could probably engraft JAK-mutated cells also at decent levels. And this is what we did, and this is what enabled us to do treatments in these animals. And I'll show you the results here. So you can see there is decent engraftment in the first place. And if you basically treat these cells for a short period of time here at week 4, the treatment starts, there's not a big difference between these animals. However, after 20 weeks of treatment, what you find is that the combination clearly shows a decrease, and this is a log-fold decrease. And this is true in the peripheral blood of the mice and also in the bone marrow. In the bone marrow, it's even a bit more pronounced. Now, one shortcut of this, or one shortcoming of this experiment is that, again, I said it before when I showed you the flow data, the cells out of the patient are never 100% JAK mutated. They may be 40 or 60% JAK mutated. So you have normal cells as well. You have normal cells plus JAK mutated cells. So one question that we wanted to ask, knowing that Claudia was able to engraft normal cells, out of these human cells, this is only human 45 that we detect in the bone marrow, how many percent of those are still JAK mutated cells? So the way to do it is to sort them out and do a chiral sequencing of JAK2-VF. And this result was even more astounding because on the molecular level, the JAK mutated cells were basically eradicated. So you can say whatever you found there was mainly leftover normal hematopoiesis over JAK mutated cells. So it seems to be efficient. This is something that from this point we thought we could take into a clinical trial. I'll show you in a second, but first one last aspect. Can we do that type of analysis, large-scale phosphoproteomics in primary patient cells? And we took that experiment on, and I did it in 2 ways, either taking the cells out of the patient, treating with the inhibitor, doing phosphoproteomics, and the second part was having the patient come in the morning, and the specifics of ruxolitinib as a JAK inhibitor is you have to take it twice daily because the half-life is only 4 to 6 hours. So you could let the patient come with no levels of rux, draw blood, let him take the pills, and 1 hour or 2 hours later draw blood again. So you have an in vivo exposure. And what you can see is this works. We can identify almost the same targets, a large amount of them, and YBX1 is clearly among them. And what drives our interest now is we started with mRNA splicing. Of course, we want to explain other cellular functions that are changed here, and what is not labeled here, but clearly a part of it is chromatin-related proteins that are differentially phosphorylated. So the last couple of slides, in summary, we were able, by phosphoproteome profiling and functional characterization, to identify proteins involved in mRNA processing as targets of mutant JAK2. And we found that one of these targets that are modified, YBX1, disrupts ERK signaling and is therefore able to induce, its inactivation is able to induce apoptosis in JAK2-mutated cells, and this can be exploited in a therapeutic manner. This is something, and I'm very happy to say so, that this concept went to a clinical trial. The ADORE trial is sponsored by Novartis. It's an open platform trial, which means you have a combination that you test, but there are additional combinations to add in the future. The combination of JAK and MAC-ERK inhibitors was added as one of these arms. We'll have a presentation probably at this year's ASH about another arm of this trial, not the MAC-ERK combination, but this is ongoing, and I'm excited to see what comes out of it. What are we doing on top of that in the future? Just 2 or 3 glimpses. One is the question, one can dissect what different driver mutations of MPNs do in this context, and the thought process at the moment is they are pretty similar. Phenotype is a bit different, but on the phosphoproteomic level, they are not. You can see what happens with a JAK mutation. This is a mutation of the thrombopoietin receptor, and this is a mutation of calreticulin, which leads also to activation of thrombopoietin receptor signaling. They are a bit closer, but there are clear differences to JAK-related signaling. This is something that helps us in our understanding of these diseases. And the second question with a more clinical impact is, I showed you now experimental data on this JAK1/2 inhibitor, ruxolitinib. We used others like pacritinib also in this study, but we will have at least four inhibitors for these patients until next year. In Germany, we have approval for rux and fedratinib. Pacritinib was approved this year in the US. EMA approval is still pending. Momelotinib is projected to be 2023 or 24. And they have different profiles. And what is known so far is that people select them based on their adverse events side effect profile. They have different side effects here: GI symptoms, polyneuropathy, muscle cramps, but there is more than that. And what we do in a translational manner, we use these data to explore it. And likewise, as I showed for the different driver mutations, if you look at these, they are all JAK inhibitors. If you look at more than 5,200 differentially regulated phosphosites, one gets an idea how different these inhibitors are regarding the downstream targets. And it's not clear if this is direct via JAK2 or indirect, directly targeting other proteins, but we'll find out. This is unpublished data, just as an outlook. And with that, I would close. Thank you for your attention. This is my group at the moment in Greifswald. This is our funding. You can see we are also supported by the German José Carreras Leukemia Foundation. We're very grateful for it. This was a great effort together with Matthias Mann and Ashok Jayavelu. Thank you for your attention, and I'd be happy to take any questions.
Thank you very much, Florian. Taking the opportunity of being chairman. A couple of questions. So did you, in addition to phosphorylation of YBX1, total expression of the protein, it matters or not for the response? Yeah. Total expression in the mutants versus non-mutants.
That's a great question. So what we have only worked with genetic deletion, right? So indeed it may be the presence of the protein that makes the difference. We were thinking about editing the proteins or leaving the structure unaffected, but to in vivo edit the respective bases to see if the function is compromised. What we can say now, and this is data I thought briefly if I should bring it, but it's very recent data. We are collaborating with Sanjay Malhotra at Stanford, and he is a great chemist who has made an inhibitor against YBX1. And we tested that inhibitor, and long story short, it recapitulates the data with a JAK inhibitor. So this leaves the protein unaffected, I guess, but blocks the function that we do not understand in very detail so far, but same effects with the same cell types.
Perfect. That was my next question. In fact, to target the protein itself, because this is a very pleiotropic protein, which does many things. And these 4 new inhibitors of JAK here, so all of them block the active site of the. In theory, this would be very similar. These are inhibitors of the enzyme of the catalytic activity. I understand.
Yes, that is true. They should be similar in this regard. They are very different in regard to half-life. So half-life rux, as I said, 4 to 6 hours. For fedratinib, it's more than 24 hours half-life. So this on-off effect may influence signaling events.
The signaling
effect. And second thing is specificity for other kinases. So they were developed as JAK inhibitors. And then there were other off-target effects described for, let's say, JAK1, 2 kinase. But what is not known is what could be alternative binding partners and pathways that are either influenced by 1 JAK versus 2, or even 2 kinase, or FLT3, as you have seen for 1. So this variation is yet unclear, and we try to connect these changes in phosphorylation at the moment also to effects on transcription. So we've done transcriptomics on the same samples, proteomics, phosphoproteomics, so we can connect basically every level from phosphorylation changes to transcription factors.
I think last one. No, I think you answered that one, yeah. That was the question. So remember that you can ask online questions using the questionnaire you have there. Questions from the audience?
Florian, very nice. I mean, we have read the paper, but still it's beautiful to get it explained. I think the main issue whenever we come to ERK signaling is that it's just so central to so many functions, so many signaling cascades, that many ERK inhibitors perform extremely poorly in terms of toxicity. And I think you also have seen that you have your 10% mixed population of cells, mutant and wild type. The double treatment kicks it down to 1%. That means you also lose a lot of non-mutated cells. So a big discussion in signal field always, if we have like one of these signaling cascades, and if we inhibit the central part, it might be very toxic, but going to the periphery could be a much more better strategy, because then the window of therapeutic opportunity might open. Do you think, I guess you will be able to benchmark this, if YBX inhibition, which is very indirect, is more permissive, or whether MKNK1 inhibition, which is only one modulatory element of ERK signaling, could be more permissive and more beneficial to have a wider window?
Yeah, that's an excellent question. So first of all, I can confirm that there's a lot of toxicity when using, especially in combination. Also in the ADOR trial, there were a lot of signals for toxicity, one has to say clearly. So YBX1 inhibitor, as I said, this was, I think, published last year, first time by Sanjay. So it's a very new inhibitor. And That would be an option. We think so. One aspect probably that may limit toxicity is the fact if you work with short-acting JAK inhibitors like ruxolitinib, it may be off in the time when it's a matter of timing also. When you expose to the MEK inhibitor, you could basically wait for the rebound and give it like intermittently. This would be an option from my point of view. This is, by the way, also the option to combine. This sounds counterintuitive, but people combine interference with JAK inhibitors. So one would say, how does that work? Because interferon signaling is depending on JAK, right? But that's exactly the thing, because the short-term, the high levels, but they decrease rapidly, and in the meantime, the interferon signal can go through and do its work, and maybe such an intermittent schedule is also good for toxicity. MKNK1, the inhibitor did not make it to clinical use, so there must be, I think, an issue with bioavailability, would be my guess.
Very nice talk, Dr. Heidel. Thank you. What about the action of JAK inhibitors like ruxolitinib for treating severe acute respiratory syndrome produced by coronavirus?
Yeah, that's an excellent question. This was used in multiple clinical trials. There were found some benefits for selected subgroups of patients, but the effect was not sufficient to gain approval for COVID-19. The reason is probably that the first trials were kind of started late, and probably once the inflammatory circuit is started and inflammation is massive, it's kind of too late to start with the medication. Probably you have to get that early on, but that's hypothesizing. I don't know what the reason is, but I think that in a nutshell, for selected populations in certain trials, there was indeed a benefit in blunting the inflammation. I was hoping for better effects because it's using it in MPN. It's impressive in this regard. It's not impressive for the allelic ratio, but if you have a patient that has, these patients have frequently massive inflammatory syndromes, bone pain, muscles pain and spasms, massive itching of the skin. And if you start a JAK inhibitor treatment, it's gone within 2 weeks. I was hoping for better effects regarding COVID as well, but it was limited.
Thank you.
Thank you for the talk. It's very nice. I have two questions. One more general about the first part. You mentioned that with time, people accumulate mutations in JAK and that in hematopoiesis, and that might affect other phenotypes beyond the hematopoietic system compartment. Yes. So I wonder if these YBX mutants develop phenotypes with age. If you keep the animals aging after a year, for example, do they develop phenotypes in addition to hematopoiesis?
That's a very good. We did not age the YBX1 mice. This is true. We looked. I think the maximum was 1.5 years, which is in the aging world not considered old. We don't know yet.
And thank you. And the second one is about have you evaluated the crosstalk between the ERK and other kinase P38, etc., and if YBX1 is also involved in others kinase?
Yes, yeah, that is true. So YBX1 is as I said, a pleiotropic protein, and I think the difficulty about it is that the context dependency is super high. So usually we are talking about is it relevant in this specific in this specific cancer type, but here even within one cancer type it may exert different functions. We have done experiments by the side in AML models that are not driven by a JAK mutation, so by other mutations. And we see that YBX1 still plays a role, but it does not exert this effect on splicing of signaling pathways, probably because the leukemias are not dependent on signaling pathways, but that it stabilizes oncogenic transcripts on the mRNA level, and if you inhibit it, these oncogenic transcripts get lost. So even that is context dependent.
Oh, many thanks.
Well, first of all, thank you. It was really interesting. My question is, there was a slide where you mentioned that this protein, YBX1, protects cells that are have JAK2 mutations, right, from apoptosis. That is correct, right?
Say it again, please.
Basically, YBX1
Yeah.
protects cells which have JAK2 mutations
Yes.
from apoptosis, right?
Yeah, exactly.
And that remind me of in breast cancer, cells which are defective in homolog recombination.
Yeah.
Depend on PARP route, right?
Yes.
Which is why some germinal line mutated breast cancers are treated with PARP inhibitors. And I saw like a comparison, and I didn't know if I was correct, like in that way.
Yeah, concept-wise, you're absolutely correct. This is basically this overcoming resistance approach with PARP inhibitors is something that targets DNA damage in this direction regarding breast cancer. We did not see any signs of an altered DNA damage response in this model system. I also think this is a JAK mutation is, unlike other very aggressive cancers, not driving massively. There's a hyperproliferation, but in terms of invasiveness or instability of the genome, that is not a real hallmark. There have been reports that that PARP inhibitors to some extent synergize with JAK inhibition. That has been reported. We did not investigate this type of pharmacologic perturbation in the context of YBX1, so I could not make any statement on that.
Mm-hmm. Okay, thank you.
Great talk. If I understood correctly, YBX1 is regulated by changes in phosphorylation at conserved sites. Are these sites known to be any known DNA motifs? Because it is also affecting localization, nutrient localization.
So you're asking for DNA binding of YBX1? Is that correct?
Yeah.
Yeah. We of course we looked into that and did chip experiments with YBX1 that did not show any direct regulation of genes related to JAK-STAT signaling and neither to MEK-ERK signal deregulation. So this was done as a control in the revision process of the manuscript, but we did not find a direct connection there. But it's known to be able to bind as a cofactor at certain sites, but obviously not in this context.
Cool, thank you.
Yes, sorry, very quick. Thank you for the presentation, very nice talk. One quick question, do we have expression data of YBX1 in these types of cancer? Yeah? So do we have any Kaplan-Meier or survival plot on YBX?
We don't have that for this type of disease.
Okay.
The reason is that well, actually, one could do that in myelofibrosis patients because you can't do it in a polycythemia vera essential thrombocythemia because the median survival is 14, 20, or 25 years. So having their survival as a readout is difficult. For myelofibrosis, it would be possible. We don't have that. We found slight differences in the AML models that we used, but it may be restricted to certain subgroups.
Thank you.
Okay, in the interest of time, we should finish here. Thank you very much again.